Hi guys, my name is Lindsay Miller and today I'm going to be telling you a little bit about my science fair project. So basically what my science fair project tested was the effect of these tiny little silver particles on a fish or like an aquatic organism called Daphnia magna. And pretty much what that is, it's just a super tiny aquatic organism, so it's kind of like a fish. And they're also called water fleas because they look like bugs. And I wanted to test the effect of these silver particles on them because these silver particles, known as nanosilver, are found in many of our common household items such as food packaging, makeup, and clothing. And recently, scientists have discovered that they have been getting polluted into our bodies of fresh water where these Daphnia magna live. So in order to test this theory, what I did was I took these little silver particles and I added them to the tanks that contain the Daphnia magna, living in pretty much their normal ecosystem. And I counted how many of the fish died because of the presence of the Daphnia magna. If when they were eating, they consumed the Daphnia magna and how much accumulated in their stomachs. And then after that, I sequenced their DNA in order to see if the nanosilver particles caused a genetic mutation. So in order to do this, I put about 50 of these small fish into a tank with the algae that they normally consume, and then I added 2 different concentrations of nanosilver to each tank, one at a pretty low concentration and then one at a higher concentration. And then obviously I had my control group, which had none of the silver particles in it. So then after I did that, I added the nanosilver to each of the tanks and I let the fish live in those tanks for about 4 months. Throughout the experiment, I counted the populations in the tanks 3 times. So I did it when I put them in there, in the middle of the experimentation, and then after the experimentation to see how many were still living in the tanks. And those results showed that in the control group population, at the end of experimentation, which was 125 days, was 113 Daphnia magna. And then the low nanosilver group population had 90 Daphnia magna in it. And then the high nanosilver group had 34 Daphnia magna in it. So what we can see from that data is that the nanosilver was pretty harmful to them because it did kill off a lot of their population. Then I also saw that in the higher nanosilver groups, there was a higher bioaccumulation because at the 380 wavelength, there should be a drop in the number, which would indicate that there was a change in the percentage that was in their bodies. So from those results, I could see that the nanosilver had accumulated within them, meaning that over time it built up in their body. And lastly, when I sequenced their DNA, I saw that there was a large change between the control group and the high nanosilver group. I highlighted the main points of the DNA segments, which are pretty much just these chains that code for what is supposed to be happening within their bodies. So a change within this would mean an important physiological change within the Daphnia magna. So as you can see, these are the main sequences that were changed on the control group into the high nanosilver group. So all these pieces that were highlighted are the ones that really changed throughout the sequence, meaning that in this high nanosilver group, the higher nanosilver, the higher amount of silver particles in the water had a greater effect on the Daphnia magna than the low nanosilver concentration did. So pretty much overall what we can learn from this is that these little tiny particles that are in our everyday human products that are getting polluted into the environment can have a large effect on these Daphnia magna, and that we as humans need to understand how we are affecting the environment, because it is very, very important to understand what we are doing with our products, since they are pretty much getting polluted into the environment, and we have to understand what kind of an effect we're having on our ecosystems and the organisms within those.